Hello, 7th standard Cbse children. Welcome back to video session 7. I hope you are doing well. Have a good day. Have a nice day. Today we are going to do video number 7, where we are going to continue the lesson chapter 2, Nutrition in Animals, as part 3. Right? We have been doing Nutrition in Animals as part 1. What is nutrition? What are nutrients? And then stages or steps involved in nutrition as ingestion, digestion, absorption, assimilation and egestion. I hope you remember that. And then we have learned the process, the structure of alimentary canal, where this alimentary canal is also known as digestive system or digestive tract or gut. I repeat, the digestive system, it is also known as alimentary canal, digestive tract, alimentary canal, digestive tract, digestive system or gut. Sorry? These are the different names for digestive system. Sorry? What are they learned? The human digestive system, starting from the mouth. I am just recollecting the points of first two videos. Video number 5 and 6. Right? What is the process of digestion? Structure of digestion. Mouth having buccal cavity where it has upper and lower jaw, heterodont, diffeodont, another milk teeth and permanent teeth. What is tecodont? I said when the teeth are inserted into the socket, the ear gum that arrangement is called tecodont. I hope you remember this. And then muscular tongue, fleshy muscular tongue with the taste buds. What is frenulum? I said frenulum is a thin fold of skin which is attaching the tongue to the floor, on the floor of the buccal cavity. On the lower bottom glass, lower jaw, buccal cavity attached. That is called frenulum. Right? And then the teeth types are heterodont, has 4 incisors, 2 canine, and then premolars and molars. I hope you follow this. I repeat, 4 incisors, 2 canine, and then 4 premolars and 6 molars. Each in upper jaw and lower jaw. So that is called dental formula. What are the types of teeth I said now? Can you repeat? Incisors, canine, premolars and molars. Say it again. Incisors, canine, premolars and molars. Are you clear? And then continuation of that is the buccal cavity. It is the esophagus or the food pipe, where it is continued as pharynx. And this pharynx, it receives several opening. What are all? First, buccal cavity opening, and then ulla epiglottis, that is preventing the food entering into the windpipes. Another opening, and then 2 nostril that opens again into the pharynx, and then a pair of eardrum, காதலோட ஓப்பனிங்கும் இருக்கு. அதை நம்ம சம்டைம் ஆன கொட்டாவி விடுவோம்ல, யானிங், அப்போ வந்து காதை அடிச்சுக்கிறது இல்லை. ஸோ அப்போ more air enters into the ear, blocks the pharynx. மறுபடியும் கொட்டாவி விட்டால் அது போயிடும் இல்லையா. that is yawning சொல்லுவோம். Okay, so that is pharynx, continued as esophagus, and then stomach, where it is having anterior cardiac end and posterior pyloric end. The body of the stomach is called fundus. I hope you recollect this. Continuation is the C-shape duodenum. U of so this is inverted C-shape duodenum, and that is forming the part of the small intestine. Now the small intestine, there are three parts where it is inverted C-shaped duodenum, and then jejunum, this cutting crossing of the jejunum, and then small intestine proper, that is called ileum. I-L-E-U-M, ileum. I hope you follow. And that small intestine is continued as large intestine. And before that, it is having a blind pouch called cecum. Okay, and then this continuation, it is the large intestine where it is having, this is ascending colon, transverse colon, descending colon, rectum and anus. This is forming the part of the large intestine. I hope you could recollect this. So this is, we have done it, video number 5. Now in video number 6, yesterday, we have learned this digestion in the mouth. What does it mean? What have we understood? As mouth contains heterodont, different types of teeth, where incisors are meant for cutting, incisors, they are meant for biting, and then canine, it is meant for cutting and tearing, where premolars and molars, they are used in chewing and making the food into simpler particles. Right? So with that, the chewed food is mixing with saliva. There are 3 pairs of salivary gland. Before that, this digestive system, the associated digestive glands are salivary gland with the 3 pairs of salivary gland, and then liver, largest gland in the digestive organ, and then pancreas. I hope you follow. This salivary gland, it is of 3 types: parotid in the cheek, sublingual beneath the tongue, and submaxillary at the angle of jaw. I repeat, parotid in the cheek, parotid in the cheek, sublingual beneath the tongue, and submaxillary at the angle of jaw. This is the location for salivary gland. And this salivary gland secretes saliva, where it contains ptyalin, the amylase enzyme acting upon starch. Right? And then bicarbonate forming a lubrication, and mucus making the food into bolus. And there is another enzyme called lysozyme, which is acting as antibacterial agent. This is saliva connection. I hope you remember. This saliva acting on starch. So the food that we are eating, if it contains this food that we are eating, if it contains starch, it will be digested in the mouth itself. And from the food, from the buccal cavity or the mouth, food is called bolus, pushed into the esophagus by the process called swallowing. Swallowing is right. That swallowing is taking place by the act or phenomena that is called peristalsis. The food pipe, it is formed of smooth muscles, elastic in nature. So when the food is swallowed, it is moving by rhythmic contraction and relaxation of the smooth muscle. That movement is called peristaltic movement. I hope you understand it. And then that bolus, that bolus is reaching the stomach. Okay, and this stomach, it is formed of anterior cardiac region. Stomach, it is formed of anterior cardiac region because it is towards the heart. So that region is called cardiac region, and posterior portion, it is called pyloric region. This portion is pyloric region, and the body of the stomach, it is called fundus. I am just recollecting. Okay. Now the stomach, it is formed of gastric glands having lining layer of gastric pits which has gastric glands. The secretion of the gastric glands are called gastric juice, which contains pepsin, renin and HCl. Pepsin, renin and HCl. Now this pepsin acting upon proteins and made into peptones, polypeptides. Pepsin acting upon protein into peptones and polypeptides. Okay, whereas this renin, it is a milk protein where it is normally found in children. Another children, பால் குடிச்சிட்டு சம்டைம் கக்கிடும் இல்லையா, அப்போ அந்த மாதிரி கேர்டு மாதிரி தயிர் மாதிரி precipitate. That is inactive form is made into active form. I hope you understand. So this inactive form is made into active form, and pepsin, renin is a milk protein. I hope you understand this. And then HCl, it is acting upon, otherwise we say HCl is making the medium acidic. Where you need to remember, HCl is secreted by a special type of cells called oxyntic cells, and it makes the medium acidic and protecting the wall also, and it is as acting as antibacterial agent. Okay, so this pepsin, HCl secreted by oxyntic cell making the medium acidic. I hope you understand this. And then we are continuing. So we say the inactive pepsinogen, gastric juice la, the inactive pepsinogen converted into active pepsin, and it acts on protein in ingested food. I hope you understand this. And then hydrochloric acid kills bacteria swallowed along with the food and making the medium acidic. And the mucus protecting the walls of the gastric layer. Are you clear? And then at this stage, the semi-digestive food in the stomach, it is called chyme. And that chyme is moving into the small intestine, duodenum portion through the pylorus. I hope you have understood up to this. Then we are learning about the digestive glands as pancreas. Pancreas, it is acting as dual gland. Rendu way it is acting as exocrine pancreas and endocrine pancreas. I repeat, pancreas is acting as a dual gland as exocrine and endocrine part. Exocrine pancreas secretes enzyme as pancreatic juice. Are you clear? So exocrine pancreas secrete enzyme as pancreatic juice. Right? And this pancreatic juice, it is acting upon the food as trypsin, chymotrypsin, carboxypeptidase, amylase and lipase. These are the pancreatic juice enzyme content. What are all? Trypsin, chymotrypsin, carboxypeptidase, amylase and lipase. What do you know? This trypsin and chymotrypsin and carboxypeptidase acting upon this protein, and it is making inactive form into active form, and then amylase. If it are any part of the carbohydrate not digested in the stomach, it will be digested by the pancreatic juice. And lipase, the fatty substances where it is digested into fatty acids and glycerol. Fat content is digested into or converted into fatty acids and glycerol. Now this pancreas acting as endocrine portion, pancreas endocrine portion as islets of Langerhans. Islets of Langerhans. You know that this laser so I am going little detail, right? Endocrine pancreas, it is called islets of Langerhans. Okay, and this islets of Langerhans. Now say this is pancreas. At anterior portion, this islets of Langerhans, and at the other surface, it is enzymatic portion. Pancreatic juice will be secreted. Now this islets of Langerhans, it is formed of two types of cells, that is called alpha cells and beta cells. And they secrete hormones. Hormones are secreted by endocrine glands, which are required in very minute quantity, and they are straight away poured into the bloodstream. So it is also known as ductless gland. For exocrine gland, it is a duct gland. In a salivary gland having salivary duct. Then pancreas, it will have pancreatic duct. Next liver padiko, adorade bile duct. So through that it is reaching the target region. Whereas this hormone secreted by these endocrine glands, they are straight away poured into the bloodstream. So they are called ductless gland. And they secrete hormones. Okay. Now this alpha gland cell secrete glucagon. Alpha cell secrete glucagon and beta cell secrete insulin. Insulin is doing a major role here. Another, if insulin is not secreted in sufficient quantity, person will be suffering from blood sugar. What is blood sugar? Diabetes. Actually, the carbohydrate that we are eating, the content of the carbohydrate, it is converted into glycogen. Okay, by various chemical reactions, it is converted into glycogen, and that glycogen is stored in the liver, right? And then by the secretion of this insulin hormone by the beta cells of islets of Langerhans, it has to be converted. Right? It has to be converted as glycogen. And if it is not so, then the level of blood sugar will be increasing. The glucose and convert panana. So if it is not so, the blood sugar level will be increased. A normal person should have the blood sugar from up to 90 to 120. If that 120 above, it is prolonged for 1 or 2 months continuously, then it is a person is diabetic. It is having sugar patient, diabetic patient. That is. So, the role of insulin is very, very important. I hope you understand. So, this is for pancreatic. This is for pancreas. Pancreas acting as exocrine and endocrine. Right? Now, the exocrine part of the pancreatic gland secrete 3 enzymes, lipase, 3 enzymes, trypsin, chymotrypsin, lipase, amylase, and carboxypeptidase. Right? And they are acting upon fat, lipase on fat, trypsin and chymotrypsin on protein, and amylase on starch. And then islets of Langerhans, as I said now, it is alpha cells and beta cells. I hope you can recollect this. Then, next one, what you are learning? One mission. Where are we? We are with the pancreas. Yeah. So, கொஞ்சம் detail ஒரு 2-3 book reference சொன்னேன்னா, it is easy for you to understand. So, process of digestion in the stomach சொல்லியாச்சு. Pancreas which is below the stomach secrete pancreatic juice. Right? It breaks down carbohydrate, protein, fat into simpler substances. Okay. Then is the liver. Then about the liver, it is as 2-lobe structure as right lobe and left lobe. Liver is a bilobed structure as right lobe and left lobe. Okay, and then it is reddish brown in color, and it is divided into 2 main lobes, right and left lobe. Right? And right lobe is larger than the left lobe. Okay? And the liver secretes bile juice, which contains bicarbonate, cholesterol, and then bile salts and bile pigments. Will this continue? So, the liver secretes bile juice, which is formed of alkaline solution, bitter to taste, which contains cholesterol, bile salts, bile pigment, and bicarbonate salts. Now, sometimes we bile excess secretion. Right? At the same time, when bile juice, when it is secreted in excess and when it mixed with the blood, it is called the condition jaundice. Jaundice for the yellow item, the yellow alkaline fluid bile juice. Right? So, this bile secreted by the liver, it is temporarily stored in a balloon-like structure called gallbladder. And from the gallbladder, it is continued as bile duct. This gallbladder is continued as bile duct. Continued as bile duct. And that bile duct joining with the pancreatic duct and both of them together forming hepatopancreatic duct. I hope you understand. I repeat, liver secretes bile juice and that bile juice is temporarily stored in gallbladder and from there it is taken to the duodenum through bile duct. And this bile duct joining with the pancreatic duct and both of them opening into the duodenum, that is as hepatopancreatic duct. Hepatic nalle concerned with the liver. Now the word hepatic is concerned with the liver. So this hepatic duct, hepatopancreatic duct. I hope you understand. So that bile juice, as I said, bile salts and bile pigments. Bile pigments are bilirubin and biliverdin, which are the breakdown products of hemoglobin. And bile salts are sodium, N-sodium, sodium glycolate and sodium taroglycolate. Sodium glycolate and sodium taroglycolate. I hope you understand. Now this liver, it has bile salts, sodium glycolate and sodium taroglycolate and bile pigments, bilirubin and biliverdin. But this bile juice, it is taking part in emulsification of fat. The bile salts help in digestion of fat by bringing about the process called emulsification. Emulsification, it brings about large fat droplets into smaller one. Emulsification as large fat droplets are made into simpler one. That process is called emulsification. I hope you understand. Now the functions of the liver I have given in the QB, you know, just listen. It controls blood sugar and amino acid level. It synthesizes fetal red blood cells. Fetal red blood cells. Now fetus, the development of the embryo. When a mother is pregnant, the first 3 months development, it is called embryo. And further development of the embryo, it is called fetus. Okay, so that fetal membrane, fetus spelling F-O-E-T-U-S, fetus. Okay, now this fetus synthesis of fetal red blood cells, then it produces fibrinogen and prothrombin, the protein content. It produces the protein content, protein type, fibrinogen and prothrombin, which are the most essential for blood clotting. I repeat, liver produces fibrinogen and prothrombin as a protein and it is used for blood clotting and it destroys blood cells. What does it mean? Now, the chapter RBC, red blood corpuscles, they are produced by the bone marrow and its lifespan, its duration of living capacity is 100 to 120 days. After that, it is destroyed in the spleen and liver, right? And then it stores the minerals iron, copper and vitamins A and D. Bile juice stores minerals iron and copper and vitamins A and D. Then it produces another coagulant. It produces another coagulant that is called heparin. It produces another anticoagulant called heparin. This is very important. When a person is suffering from heart attack or the heart problem, in the heparin secrete blue eye. And this heparin should be produced properly. So it is an anticoagulant. Anticoagulant blood clotting, coagulator. So this heparin is acting as anticoagulant. Then excretes toxic and metallic poisons. Go through pattern. It excretes. Go through pattern. Go through. Can you? It excretes toxic and metallic poisons. Then it detoxifies substances including drugs and alcohol. Bile juice detoxifies substances including drugs and alcohol. I hope you understand this. So I repeat, controls blood sugar and amino acid level, synthesizes fetal red blood cells, produces fibrinogen and prothrombin used for clotting of blood. It destroys RBC. It stores iron and copper as minerals, vitamin A and D. It produces anticoagulant heparin and it excretes toxic and metallic poisons. And it detoxifies substances including drugs and alcohol. I hope you understand. Up to this is the liver. It is for liver. So now we say the function of bile juice. Adhe pancreatic juice, trypsin and chymotrypsin acting upon protein, amylase on starch, lipase on fat. So in the product digest reaching the duodenum. And from the duodenum through the jejunum, it is reaching the small intestine. Right? And the small intestine, it is also secreting intestinal juice. Small intestine, it is also secreting intestinal juice as succus entericus. As succus entericus. This is enzyme, intestinal juice, enzyme as amylase, lipase. Can you guess? Succus entericus. It secretes intestinal juice as try lipase, maltase, sucrase and sucrase, lactase, maltase and lipase. Right? So jejunum constitutes 2/5 of the small intestine. It starts from duodenum and ends with ileum. Ileum is the small intestine proper. The secretion of small intestine is intestinal juice and it secretes the enzyme sucrase, lactase, maltase and lipase. Okay. Then these small intestine, ileum portion, the small intestine, ileum portion, it is having a structure called villi. Numerous finger-like projections called villi. And this villi, it increases the absorptive surface area. highly coiled structure போடுவோம். But when we see inside a single, இது பார்த்தீங்கன்னா it will be like this, where the cells are arranged like this. Okay, and they are nothing but blood capillaries. And along with that, there is one more as lactate. This is called lactate. Okay, so if at all, if any of the food components are not digested, that will be completed by succus empiricus. So the completion of digestion, that is taking place in the small intestine, and then it is absorbed, the process absorption. Okay, so first ingestion mouth la mudichachu, digestion வந்து இந்த mouth la irundhu intestine varaikkum, small intestine varaikkum, completion of digestion, right? And then in the small intestine portion, that is ileum, that absorption is taking place, and that absorption to occur, it is as villi. Villi are the finger-like projections meant for absorptive surface area. I hope you understand. And then along with that, a structure called lactate. A structure called lactate, that is also mingled along with the villi. Lactate is also mingled with the villi. So after complete digestion, the complex carbohydrates, they are converted into simple sugars like glucose. I repeat, complex carbohydrates are converted into simple sugars like glucose, and proteins are converted into amino acids, and fats are converted into fatty acids and glycerol, and that process they are absorbed by this process of absorption. I hope you understand. So the wall of the small intestine has tiny small finger-like projections called villi. ஒரு இது வந்து villus. This increases the surface area of the wall of the absorption of digested food, and this villi are nothing but richly supplied blood vessels, and they absorb nutrients by the processes diffusion and osmosis and active transport. Diffusion and osmosis, they are simple process, and active transport are this passive transport, right? They absorb nutrients and transport them to different parts of the body through blood. Water is absorbed in the large intestine. I hope you understand. So this is for ileum villi portion. This is process of absorption, and then it is assimilation. The absorbed food has to be synthesized. That process is called assimilation. How the food that we eat used to by the body. After the nutrients are absorbed, they are carried out to each cell in the body by the bloodstream, and each nutrient is used in different way. The process by which absorbed food is utilized by the body is called assimilation. Right? The process by which the absorbed food is utilized by the body is called assimilation. I hope you are listening. So the oxygen we inhale helps to break down glucose into water and carbon dioxide. Listen, oxygen we inhale helps to break down glucose into water and carbon dioxide, and it releases energy, right? So the circulatory, respiratory and digestive enzymes work together to help us to get energy from the food. Then they absorbed amino acids, they are converted to proteins and help to repair old and damaged or worn out tissues. They are also needed for growth. So I repeat, the absorbed amino acids are converted to proteins and help to repair old and worn out or damaged tissues, and they are also needed for growth. Is proteins. Okay. Then the fatty acids and glycerol are remade into fats, which are either used or stored and act as energy reserves. So this is how after absorption it is reused, resynthesized, and that process is called assimilation. Okay. And then about large intestine, as I said, cecum is the blind pouch, and when it gets inflamed or accumulation of fluid, it is called appendicitis. And then this process, last step in the digestion is digestion. Now the part of the food that is not digested or absorbed enters the large intestine. There they absorb water and salts. Right? So the water and salt, they are absorbed by large intestine. Excess water absorbed by the blood vessel is taken to the kidneys to be removed with the urine. Just a minute. The last step in the digestion is digestion, the part of food that is not digested or absorbed enters the large intestine. Okay, and it is which absorbs some water and salts. Excess water absorbed by the blood vessel is taken to the kidneys and to be removed along with the urine. And the remaining undigested and absorbed material is stored in the rectum until it is thrown out of the body through anus in the form of feces. Feces na motion, and this process is called defecation. The process of removal of undigested food is called defecation. And then, so this large intestine, as I said, the unabsorbed and undigested food is passed into the large intestine, and then it has cecum, colon and rectum. The cecum is a small blind pouch situated at the junction of small and large intestine. So the blind end of fingerlet structure, it is vermiform appendix, where it is a vestigial organ. Right? And then colon and the ascending colon, transverse colon, descending colon, continued as rectum and anus. And the undigested or unassimilated part of the ingested food material is thrown out from the body through anal aperture or as fecal matter, and this is known as digestion or defecation. I hope you understand this. So this is the process of digestion in human. We have learned the structure of alimentary canal and the process of digestion in man. I hope you have understood. So this is our video number 7. Part 3, Nutrition in Animal, Part 3. Next, tomorrow's video session, we will learn about nutrition in amoeba and then nutrition in ruminant animals. That we will continue tomorrow. Have a nice day. Thank you.